Hey guys, we're going to try to get chapter 12 in here too. So bear with me. We're going to share screen here. Okay, you should be able to see this now. All right, this is a tough chapter here, so bear with me. We're going to try our best to go through this one. But this is one that's got some huge questions on it, including a big 10-point question on your final, probably or more, at least 10 points on describe protein synthesis. So I'm going to do my best and try to run through this. I've got about 30 minutes before I have a class. I'm going to try to hit the high points, okay? All right, so this is one we're going to look at the genetic material, how genes are expressed, and of course the big ones here is gene expression involves two concepts here, transcription and translation. Okay, so this introduction commentary here is kind of cool here. All living things on Earth have only 4 bases in the DNA: adenine, guanine, cytosine, and thymine. These bases code for all biological material, and he makes a comment here about this MC1R gene that contributes to skin, hair, and eye color is found in the nucleus of every living cell so far on Earth. So, and it determines melatonin levels, etc. Okay, kind of cool, kind of interesting. All right, all right. The first one here is we got to figure out. There's a big argument at the turn of the last century, the 1900s, as to what the genetic material was. Okay, and one of the first experiments here was Griffin's investigation into Streptococcus pneumonia. And we actually have I think we have a picture of that. Let me show you the picture of that. So here is Griffin's transformation experiment. He had a capsulated, injected live S strand that has a capsule and it kills the mice. But S strand capsulated means it has a protein capsule around the outside of it. Okay. If you inject an S strand that has no capsule and there's a little different strand of bacterium there, okay, then no capsule and the mice do not die. If you heat the S strand of bacterium here that's capsulated and kill it and inject it into the mice do not die. But if you inject the heated, dangerous, this S strand but killed, inject that into the mouse and then inject the non-contagious R strand, then the rat dies. Okay, so the inject live R strand, that's the one here that's not going to be, doesn't normally kill it, but it somehow transforms that DNA we now know into their genome and kills the mice. And so he said that there's some type of transforming agent here, and that was the point of his experiment here. He concluded that the violence could be passed from a dead strand to a non-violent living strand, and he called that transformation. Later on, they discovered that the DNA is the transforming substance. Inside that DNA from the dead cell is incorporated in the genome in a little bit. We still didn't know about that. And that brought us to a big argument that was, what is the genetic material? Is it DNA with these 4 bases, which seems highly unusual that you have a language only 4 letters? So think about the English language and all. Or would it be these amino proteins are also in DNA? And there are 20 different amino acids that give you more variety. Now, we now know that the protein is those histone packing proteins that are not the genetic material, but we now know it is the actual DNA that is the hereditary material. So we had to do some experiments to figure that out. So look at that. She kind of goes over some of the requirements for genetic material. I'm not going to bore you with all that. Y'all can read that. But the big one that's going to do this one is going to be, that's looking at Griffin's experiment again. But then we had Avery, McClendon, and McCarthy also scientists argued that DNA lacked variety, enough variability to do that. And so they were pushing the DNA, that it was these enzymes, these proteins that was the deal. And that brings us to the Hershey and Chase experiment using a virus that attaches itself called bacteriophage to bacterium, okay? Okay, and take a look at what they're gonna do. Let me show you their experiment. It's very amazing here. So they have bacterium, they grow in their beaker here, and they have a virus, okay, that they can attack. It doesn't look like the alien in some sci-fi movies you might have seen. What Alien? I think this is the one that looks like it sucks on the guy's mouth. And Alien and Aliens. Okay, they inject something in, and then what happens? Normally they put the virus on, and then it makes baby viruses. Okay, so here's what they did. They noticed there's 2 things that make up the virus. It had this DNA inside of it, okay, that they could radio label with phosphorus. Okay, they let them ingest on the bacterium. Then they take a blender, knock off the viruses, and then the radiation was now in the bacterium. That told them that the DNA was transmitted, and then they are going to produce baby viruses. Okay, she doesn't show that. Okay, but the viruses are also going to be produced and it's labeled, you know, yellow here, and then the viruses are going to be produced are going to be radioactive. Okay, baby virus is radioactive here, which means it was the DNA that was transmitted. And then he did it with the other main component of the virus is this protein shell that they can put radioactive sulfur in. They let it ingest, let it do its thing, take the blender, knock it off, and the radiation stayed with the liquid and not in the bacterium. And the viruses produced by the bacterium are not radioactive. That's telling us that it was the DNA was the hereditary material. And that is a world famous big time experiment that brought us to the conclusion of that. Okay. Okay, so you can go and talk about that, talk about how the transformation is used in genetic engineering. They can move this jellyfish gene to make it glow in other things, use it as a marker, blah, blah, blah. Moving on. Now, we get down to the big one here. DNA then is the hereditary material based on Hershey and Chase experiment. We have two nucleotides that are purines called adenine and guanine, and two that are pyrimidines, thymine and cytosine. Okay. Now, Sharkov was one of the famous Nobel winner of Europe, and he determined that the amount of A, T, G, and C in DNA, A and T is always equal, and C and G is always equal, suggesting that these two are maybe complementary pairing to each other. Each human chromosome contains an average of about 140 million base pairs. The number of possible nucleotide sequences then is 4 to 140 millionth, which is crazy to think about. Okay, and what he determined then was that adenine and thymine, adenine, notice the double ring, thymine bind to each other and their amounts are equal, and C and G are equal. And he looked at humans, fruit fly, corn, fungus, and a couple of bacterium, and he saw that. So A and T equal, and A plus G equals T plus C. So there was some pairing on, and we're going to find out that our heroes today are going to be Watson and Crick, and they come up with the structure of DNA, and become two of the most famous scientists of the last century because they're credited with showing and describing the structure of DNA, which 10 years later they won the Nobel Prize for that with Maurice Cheeks, who also did this work with Rosalind Franklin, who's one of our first women we mentioned here in science here. She was studying what's called X-ray diffraction of DNA, where you take X-rays of DNA, and she found that it had a concentrated various solutions of DNA is made. It can separate into fibers under the right conditions. Fibers can produce X-ray diffraction, and this can tell us the structure of the DNA. And she was able to tell from her X-ray diffraction photography the DNA is a helical, and some portion of the helix repeat itself. A colleague of Franklin's, Wilkins, showed her photo to James Watson, who understood its significance about the structure of DNA. Okay. Now, the thing I think is super crazy cool about Watson and Crick, Watson is an American who's still alive. Crick died a few years ago. He was also brought back in to complete the Human Genome Project, where we mapped the entire human genome some 15, 20 years ago. So he's still alive to my knowledge. He has not passed away. He's close to 90 now, I think. But he was very young when he did this. But they did not do any actual scientific experiment contribution to the data to this thing. Well, what they did is what I think I love about some of our American thoughts of our country is they took all the work everybody else did, locked us up in a room, and from the proven work that others had done, they were able to be the first people to construct a structured DNA that we'll see in just a second. I think that's really cool. There's even been a movie called The Alpha Helix, and there's been many books written about it. There's even talks that maybe I saw an interview with Crick right before he died, and I think he had a picture of Rosalind Franklin in his office. I think there's some little romance there between them. Maybe it wasn't Wilkins. Maybe she gave it to, I read in other books that maybe she gave it to Watson. But anyway, this is what the X-ray Rosalind Franklin picture here, and this is showing what this represents. The X represents that it is spiraling helical structure, and also the black lines here I've read in other books indicate lots of hydrogen bonding holding these pairs together. So they knew they had a spiraling staircase, alpha helix structure, and then Sharkov had A equals T, and A plus, A and C equals G, and so and purines equal pyrimidines. So from all that, they were able to construct this structure in 1953. They won the Nobel Prize in 63, I think, or 62 actually. There it says in the PowerPoint. And one of the great historical questions is Watson and Crick won the Nobel Prize 9 years after publishing their model of structured DNA. Wilkins, Cheeks also was with them. Yes, Franklin, Rosalind Franklin's co-worker, Wilkins won the Nobel Prize for Watson and Crick. And people always go, why didn't Rosalind Franklin win it? She had died of cancer since then. And no, they do not give the Nobel Prize to someone after they're dead. She would have been the first woman, I believe, in science to win the Nobel Prize if she had lived that long. They did have an instance like a year or two ago where a guy got it, had a heart attack, and died. They did go ahead and give it to his widow, okay, or his family, since he was alive when they announced it. I think that's a nice trivial quick question here. So DNA then is a double helix model similar to a twisted ladder. Sugar phosphate backbones make up the sides, and hydrogen bonds make up the rungs or the steps in the ladder. The 2 DNA strands are anti-parallel. Okay, so here's what it looks like. There is Watson and Crick in 653 with their model of DNA made out of pieces of metal and tin that's sitting in a museum today. I think that may be in the Smithsonian or one of the great museums today. Watson on the left here was barely in his 30s, and Crick was older. Crick had actually helped create the nuclear weapons in England. He was an Englishman. So he thought he'd get in something that would be less dangerous. Oh Lord, did he not know that his discovery of DNA would lead to genetic engineering and all the dangerous things we can do with that. So some of the worst things we have. But you can see here, A to T, you get the spiraling staircase. Look at that. You got to draw me some of this up in complementary base pairing. Notice that C and G, cytosine and guanine have 3 hydrogen bonds. Adenine and thymine have a pair of hydrogen bonds hold it together. So hydrogen bonds hold it together. I want you to draw some of that on the test for me, and or describe its structure, and then we'll talk more about that. DNA replicates by semi-conservative replication, and they also suspected this. So it splits apart in A to T, C to G, and you end up with 2 new strands that are half old, half new, and this is very semi-conservative, meaning that all the new ones are half old, half new, and the chance of error goes way down there until you have less mutations. So they not only did they describe the structure of DNA, they explained how it could replicate during interphase, if you remember. So replication requires an enzyme DNA polymerase. It's going to be the one that causes it to copy itself. This is now used in genetic engineering to make more copies of DNA at a crime scene, right? PCR, polymerase chain reaction technique. Prokaryote replication, a little bit different. Eukaryotes, telomeres at the end, remember, also have to be replicated. They're going to fall off every time if you don't have these enzymes. Telomerases can add more to it, but after a while, remember, this helps stop division. These telomeres, maybe the fountain of youth is at the end of a chromosome called a telomere. So every time the cell divides, a piece of this falls off. Okay, and then when telomeres run out, then they stop dividing again. Okay, and there are enzymes that can regulate this. So cancer a lot of times is errors in these enzymes, telomerases, that add more telomeres because cancer cells appear to live forever. Here's the eukaryotic replication, right? And then here is the eukaryote. So remember, they have that circular piece of DNA. They do it that way. Enzymes, and here we go, involved here, a couple of more to it, DNA polymerase. We also have a concept called Ozaki fragments here. As it separates, this side is continuous, but because of the anti-parallel strands on each side of it, then these fragments have to be filled in. They're called Okazaki fragments. So you have multiple, this one has to go this way, and then there's another strand that's been left open there. Okay, and there's some cool pictures of that. Accuracies of replication, look at that, 1 error per 100,000 base pairs. And then they have the capacity to identify and correct those errors. Okay, then the next thing is, what does all these nucleotides represents? Okay, Bill and Tatum here suggested a 1 gene, 1 enzyme hypothesis, right? And the flow of genetic information then goes from DNA to RNA to protein production, okay? And so we're still figuring out, we'll talk about how we control this gene expression of this, but this is very big. The central dogma of biology is DNA is copied into RNA, and every 3 letters in DNA code for an amino acid or a stop or start codon, and every 3 letters then is a codon in the messenger RNA that code for a particular amino acid to go in position. Think of proteins as boxcars on a train track, and the right order makes a certain protein. A different order makes a different protein. And remember now, our enzymes catalyze everything in the body. So DNA tells us how to make proteins, and proteins do all things. Okay? So DNA is transcribed into messenger RNA. The messenger RNA then goes to the ribosome and the cytoplasm, where we translate that code of the codons into a sequence of amino acids. And that is the key central dogma of molecular biology. So important. DNA is copied into messenger RNA. The DNA never leaves the nucleus. The messenger RNA goes out to the ribosome, and we'll do translation there. Okay, there are several players here, as you can imagine. Remember, RNA has uracil in the place of thymine, so we don't see thymine in RNA. RNA also has ribose sugar instead of deoxyribose, so there's a sugar difference. There are 3 types of RNA that are made at the nucleolus in the nucleus, and they include messenger RNA, that's going to be the copy. Okay, ribosomal RNA, that's going to go out and form the ribosomes in the cytoplasm. So 2 subunits snap together, and then there's a transfer RNA that brings the amino acids to the ribosome to build the protein. Okay, here's messenger RNA, a single strand, okay, with uracil in place of thymine. Here's another little more comparison, different sugar, uracil in place of thymine, RNA is single-stranded, DNA is double, and there is no helix in RNA, it's just a single strand, DNA has a helix. Okay, now, so I think one of the bigger questions then is, what is the secret code here? Now, how many of y'all remember what I mean by secret code? Back in my days, there was a cereal had toys in it, and there was a cereal and Captain Crunch toy, and the cereal Captain Crunch toy was the Captain Crunch decoder ring. And the decoder ring was just a little plastic ring that basically had a little thing you click, and if you wanted A, its number, it had a number associated with a letter. And if you and your friend had the same color ring, then the sequences were the same. So in other words, very simply, A is 1, 2 is B, 3 is C. So if I want to send you, I love you, Lisa, it would be a bunch of random numbers that you'd have to decode. And so in the language of DNA, there is a code. And the code is every 3 letters. Now, obviously, there are 20 amino acids, and there's 4 nucleotides, 1 to 1 wouldn't do it. So they did 4, 1, that ain't work. Every 2, you can only come up with 16 sequences. In other words, you can take U, A, C, and G. You can combine those into 16 ways, looking at 2 at a time. That's 4 to the 2nd power. And so then they said, well, maybe every 3 letters, then that would be 4 to the 3rd power, and that would be 64 combinations. And sure enough, in the genetic language of, and by the way, Nobel Prize for people figuring this out, they were able to create messenger RNA sequences and drop it into a cell extract that had everything needed to do translation. And they would put in, 15 adenines, and you'd end up with certain amino acids, 5 amino acids coming out. Or 15 of them would be 3 times 5 amino acids in a row. They would all be the same one. And after they figured it out, this is what they come up with. This is genetic code for humans. In other words, as I was saying, if the messenger RNA had a TTT or UUU, it would be phenylalanine every time. And if it's UUC, it was phenylalanine also, but UUA got leucine. So there's 2 for phenylalanine. Looking at leucine's got like 6 of them. 1, 2, 3, 4, 5, 6. Okay. And so this is the genetic code for messenger RNA, with the first letter being U, C, A, or G. And then second letter come across, and then third letter. And you can kind of quantitate it. You would never have to memorize this, but I do think it's kind of cool. A couple things that's super crazy about this, and it took them quite a while to figure this out. That all proteins begin with AUG messenger RNA that creates methionine as the first protein amino acid in all proteins. So that's God's start sentence here, start codon. And then there are 3 that end all messenger RNAs, and they stop building the protein. These are God's end of sentence period to stop translation. So 3 codons are the stop codons, and 1 methionine tells us where to start. When this was figured out, this is probably my favorite biology fact of all of biology. Then they saw the potential here, and the question then was, what about dogs and cats? What's their secret code? What about pine trees and butter beans and corn and cotton and everything else living? Well, come to find out, this is the genetic code for life on planet Earth, not just humans. And because of this, we can do genetic engineering, where we can move genes around and have them expressed in other organisms, because the basic central dogma of biology is DNA is copied in messenger RNA. Messenger RNA uses this secret code to tell which amino acid is made by the transfer RNA coming in, binding anticodon to codon, and they bring these specific amino acids based on this code. I think we need to go to the moons of Eupora or Saturn or wherever, and we need to find life in our universe, in our solar system. I'm pretty convinced there is. And when we do, if they have this code, then they are our cousins. If they have another code, it'll be the most valuable industrial information known in the history of time. And whoever gets that first will be very, very powerful country for a long time to come, because this is how we do life on Earth. What if we had a different way to do that? A different codon, a different sequence of building things that could be extremely useful and dangerous too, if you think about it. Okay, so This universal code is universal with a few exceptions. All organisms use the code the same way. They encode the 20 amino acids in these 64 triplets. Okay. The 64, then there's redundancy in some of those, right? Most amino acids encode for 2 or more codons. None of the codons copy for 2 or more amino acids, so they're all specific. Okay, and then we had our ending punctuations here. So in transcription, we're going to copy the DNA into RNA, into the single strand RNA, right? And this is pretty cool. The enzyme that does this is RNA polymerase. It's going to come in, and you're going to make the copy of that. Now, guys, how it knows which side to copy is still one of those things we're figuring out. This is one of the areas of gene control, or gene expression control. Isn't that really cool? So it comes in, makes a copy of it. And she's going to go into this quite a bit more detail than I want to. We have these pre-messenger RNA is modified. You got to put these caps on the 5' end. The cap is a modified guanine nucleotide. It helps the ribosome determine where to attach when it begins translation. Poly A tail, 150 to 200 adenines on the 3' end. Right? We talked about that, the 3', 5' end of the DNA. Okay. Once it's been produced, then there are introns that have to be cut out, and exons are the pieces that need to be expressed. So we have RNA splicing, spliceosomes that come in here and cut the introns out. Look at this. So here's the messenger RNA, and those introns have to be cut out. And once we cut them out with these enzymes, we have a product now, messenger RNA, that's had the introns removed. So introns are kind of a weird thing that are pieces of DNA in the gene that do not have to, they cannot be, has to be taken out before we can go down and do translation. That's pretty wild. Then in translation, and it talks about putting the genes for fluorescent protein into one. Translation, we have those transfer RNAs that are going to have, there are 64 of these that carry the specific amino acids based on the genetic code. And they're going to go and bind the specific amino acid, and they're going to have an anticodon that binds to the codon. Take a look. So here's what they look like molecularly, but here's how we draw them. And this end carries the amino acid, and there's the anticodon that would bind to what? CUU codon and messenger RNA. Okay? And so what happens is it comes in, brings the amino acid in, leucine in, and it binds there. Okay? And then look what happens. Then the next one comes in, and it keeps adding and building that protein as it reaches down there. It's in other words, it just keeps bringing one in, and the ribosome slides down until it hits the stop codon. That's an electron microscope showing, 6 ribosomes stuck on one messenger RNA. You have initiation. Here we go, showing it again. I'm going to go through this kind of quickly. Y'all can look at this on your own. Then it brings in another one here. See that now? Look, that amino acid is going to stick right here, and that's how the protein is built. See that? And it just keeps going until it hits the stop codon. Isn't that cool? There's that sticking to valine now, and now we're ready for another one that's going to read one more. And when it hits the stop codon, then it stops. So here's an overview of this very amazing thing, and this is how we do biology. In other words, my DNA said Robin would be ugly. I had messenger RNA that came out and made enzymes that made Robin ugly. So you guys had better genes than me, made you cute, or whatever your situation is, and there are 23,000 genes. So everything that's personality, everything that is of a genetic material, we can talk about that later, is what all that might be, is achieved through this process. Absolutely, amazingly, super important process. Okay, a little summary of it there, and I think that's it for today on that one. Y'all have a good day. We'll stop. See y'all later. Keep it going.